recorded. And I see that Chelsea just hit the recording button, so it started. And so if you missed something, don't worry. This will be back. We'll get this posted up there in that Teachable site as well for you to go back and view at any time. Okay. So I also just want to make sure during the time that we were getting logged on, you saw a screen with a bunch of sponsors. And we just want to make sure we give thank you to those sponsors that are supporting not only this meeting, but the entire series of winter meetings that we've done this year via Zoom. We really appreciate their support. Sorry that we couldn't be all be together for this, but we'll get some good information out there. And what's nice about Zoom is that we can bring in some speakers that we normally wouldn't have been able to do, which is my introduction for our first speaker. So our first presentation is called Managing Sweet Corn Worm Pests and Insecticide Resistance with Old and New Chemistries. And our presenter for this topic is Dr. Tom Kuhar, who is a professor in the Department of Entomology at Virginia Tech since 2001, where he has a 75% research and 25% extension appointment focused on vegetable and turf entomology. His research focuses on investigating the biology, ecology, and control of arthropod pests, where he then uses this knowledge to develop and deliver useful pest management information to ag, clientele, and the scientific community. And today, I've asked Tom to discuss his most recent sweet corn insect management programs with us, with an emphasis on which insecticide products are working the best, as well as reducing insecticide resistance with rotating through the different chemistries. So with that, Tom's got his presentation up. I'm going to turn the program over to Tom. It's all yours.
Okay, well, thank you, Chuck. I'm assuming everyone can hear me, and sounds like the slides are up there. So yeah, appreciate the opportunity to speak with you on an insect that I know you all are in New York, but we are battling a very similar pest down in Virginia. And in fact, it's probably not a bad idea to hear from someone who's battling it further south, because with this pest, in the warmer climates, you tend to have more and more pest pressure from it. So I'm going to share with you in the next half hour here just some of the things we've been doing over the last 10 years or so, and especially most recently, because things are changing with this pest. So just getting into sweet corn, it's very possible where you're at in New York, you've seen any of these pests attacking your corn ear. The top left, you got European corn borer. That's historically been a big problem in the northeastern US, but not as much so in recent years. We've seen a drop off with this pest, mainly from the widespread use of Bt transgenic field corn. It's caused a overall reduction in the population of this insect, which is really, really susceptible to the Bt corns that have been out there. And we've seen kind of a drop off in what this used to be. To the far right, top right, is fall armyworm, another one that could get in the ear. This is very sporadic. It's a tropical insect, so not one that you're going to get every year in the Northeast, but we occasionally will get it moving up the coast. It comes up on some storm front winds, blows the malls up here, and it comes here late. So if you got some late corn, it's possible you can get this insect. Again, it's kind of sporadic, depends on whether we get it or not. It's the same for Virginia. The bottom right, you see the brown marmorated stink bug. This is definitely a pest that it's very patchy. Organic growers seem to have dealt with the worst problems with this, but I've never seen stink bugs in the sheer numbers get on sweet corn than we have since this invasive pest has come in. You see the damage it does to the far right corner there. That's piercing, sucking, feeding mouth parts. But again, it's kind of sporadic. It's not every situation. It's often got to be near some wooded borders where there's trees. You end up getting this, and it's a kind of an edge insect. And then the other insect is sap beetles. You see to the left there, both the larvae and the adults of these little tiny beetles that'll also get in the sweet corn ear. So these are probably familiar to many of you. Just want you to be aware of them, but without a doubt, when you're talking sweet corn management and pest management, that this one is the main driver. You may get these other things, but this thing, it's pretty likely you're going to get it every year. It's just how bad you're going to get it. This is corn earworm, and focus of this talk. So it's not just me saying that those other pests are sporadic and corn earworms kind of more important. There's some pretty hard data. What you're looking at here is several locations where this is Dr. Galen Dively from University of Maryland that heads this project up. And we have sweet corn sentinel plots where we're evaluating different BT transgenic varieties, and at all these locations, you see them across the country. And just what I want you to take from this is just the black is percentage of ears in those plots that had corn earworms in them. And the red is European corn borer, blue is fall armyworm. And really what you see is just an absolute domination of corn earworm wherever you go, being the dominant pest and a very severe pest in 3/4 of all the fields across the country. The only ones that don't get corn earworm are the farthest north sites that we have in this whole project. So we're talking corn earworm as a major driver of pest management, something you probably already know. So let's get into it's important to understand its biology, and not just because I'm an entomologist and I want to spout off about an insect and its biology, but because understanding its biology can help with knowing why maybe management didn't work, and maybe some things that you might do. So hopefully many of you know it's a moth, about an inch long, beige in color. You see the picture of it there. So moths will be feeding on flowers. One thing we do know is that female moth, when there is fresh silks from sweet corn, that is the #1 place she wants to drop her eggs. Little tiny spheres you see in that photo to the left. So she will prefer to lay eggs in sweet corn over anything else if it's fresh silks. So it's basically a magnet for corn earworm. Those eggs don't take much time to hatch, a couple days or so, and that little tiny larva is going to crawl down that silk and into the ear. Once it's in there, it's protected. Often predators can't get it, insecticides can't get it, and it's important to do the control or make sure your control measures are taking it out before it gets inside the ear. So many of you know that part of it. Once it's in there, the larvae will go through several molts. By the way, it can be all kinds of different colors. You've probably seen this. Corn earworms, anything from black, red, green, yellow. It's all the same species. Just a lot of variations, especially when they get large, in their colors. Eventually, they will reach that last instar, the larvae, and then kind of tunnel out of that ear and down into the soil where they'll pupate. And you see on that bottom slide the exit hole from the corn earworm that basically grew up inside the corn ear. So that leaves behind a huge mess, and definitely we don't want that for our sweet corn. So obviously we're controlling this insect. One thing that's come into play is understanding some of its genetics and problems, and we're going to get into this in a minute, is overwintering. And there was research done long ago that shows that you're probably not getting overwintering a corn earworm where you're at. You're north of the 40-degree latitude line. That's too cold. These corn earworm pupa, which are spending the winter, they're susceptible to freeze in the soil. So you're basically not getting corn earworms very likely overwintering in New York. In Virginia, I'm probably getting some. The big concern we're having is that the work done on overwintering was done long ago, and that we believe the climates are probably have changed this equation a little bit. So I think we're getting a lot more overwintering further north than we used to, and that's causing greater problems and some greater issues when you start to talk about resistance to insecticides and things. So because it overwinters south, we're getting a kind of a migration of moths that are coming up during the season. So it's very, very valuable to monitor for moth activity. It at least lets you know they're here, they're active, and they're reproducing. And there's blacklight trap networks. You see in the bottom right, that's a blacklight trap. I know that's really big in New Jersey. And you basically, moths will come to lights at night and you count them the next day. And it at least lets you know that these moths are active and how many are out there. You can put pheromone traps on your own farm. There's different types of wire mesh in the top right and kind of a softer material mesh heliothis trap you see to the left. Either one of those you can bait with a commercial pheromone lure that's attractive to the male moth. he thinks he's mating. You could draw in males from all over, and they're coming in and they get caught. And that tells you you've got corn earworm mating going on, and that's alerts you to you're going to also have females laying eggs, especially if you've got sweet corn ready. So it's a good way to monitor that moths are here, and we got to protect our corn. So that's kind of how that's used. Here's just an example of 3 decades of doing blacklight trap monitoring in Virginia, Eastern Shore of Virginia, and just shows you how consistent corn earworm activity is. We don't seem to have a lot of it until you get to mid to late July. This is Virginia. Probably a little bit later. Chuck can maybe chime in on when activity really picks up in New York, but I'm sure it's not too much later. But the point is, there's not a lot before then. So if you can somehow get corn out of the ground, harvested, before you get to that mid to late July, your corn earworm problems certainly are not as bad. Nowhere near the severity that you're going to get as you get later. And a lot of growers know that. The later you get in the season, the more corn earworm pressure you're going to get. And the data just show this clearly. So that's a good piece of information. That was Virginia. This is Maryland, just to show you that there's very similar trends. They tend to have a lot of activity in August, as you see there with the different locations in Maryland. But also pick up on something. There's a peak that occurred right there between June and July. You see that? So this is 2020. This is last summer. And this is getting at what we're thinking, that there's probably a lot more overwintering moths further north than we were used to having. And that is very likely that peak you see there, moths that spent the winter in Maryland, and they emerged as a moth come springtime. So, and then by early summer, you were catching those. So that means maybe things might be changing with the seasonality of corn earworm. So important things for just monitoring. We're monitoring is very, very valuable for those reasons. Okay, let's get into some BT transgenic sweet corn. And I know my speaker after me today is going to focus a lot more on some of the genetics and traits in sweet corn. And but we have been evaluating these. So I'm going to tell you what we've found in recent years and what's going on with BT transgenic sweet corn. And just in my career, I've seen since the late 90s till now, the number of varieties and the types of BT sweet corn has just gotten prolific. There's a lot of options out there, a lot of different varieties, and a lot of ways to go with BT sweet corn than we've ever had. And we're going to kind of get into that. So fortunately, we've just published a paper. Galen Dively from Maryland is the lead on this whole project. It's got a lot of researchers who have looked at all these different BT transgenic sweet corn events in our trial plots. And this is basically a summary of how well these transgenic corns are performing. And just to show you, testing in a lot of different places. There is a site in New York, as you can see there. Actually, two, one out on Long Island. Dan Gilreen has got some BT sweet corn plots there. So a lot of plots, a lot of data. And the good news is that we're getting the same results everywhere. So wherever that you test them, we're kind of getting the same results across everything. First thing is, with regards to European corn borer or any of these caterpillars that may bore into the stalk, and that used to be a big deal in our corn, we're getting 100% control of that. And the BTs work really, really well at keeping borers out of the stalk of corn. But that's green tissue. That's not our major concern with sweet corn. We're trying to protect the ear. But just to let you know, any of the BTs are going to have very, very clean stalks. They're not going to have holes in them. But how about ear protection? Well, if you go back to 96, that was the first BT sweet corn that came out from Syngenta, the attribute corn it was called, and had a gene in it, Cry1AB, that coded for an insecticidal protein. So that was the first one that they had, the first one that growers were using. And it had a little bit of tolerance to glufosinate, so a little bit of herbicide tolerance. And there were a few different types of varieties that came out, sweet corn hybrids. So let's look at how that has performed over the years. So 96, it came out. And trials were being run at 26 locations and have continued to be testing that variety into last year. And now we have over 100 sites. We used to get about 82% control of corn earworm with that original variety back in the day. That was outstanding, made it very, very worth buying BT sweet corn. And but look at what's happened over the years. We're only getting about 7% protection anymore. So we're virtually getting, 93% damaged ears in the really heavy. So that's just pretty much not working at all. Problem is we've gotten resistance development to that protein in corn earworm populations. It's not killing it anymore. And it's pretty clear with those data. Moving on to another BT sweet corn, the performance series. So these were released by Monsanto or Seminis. These are another kind of sweet corn, offered a lot of different varieties and some different genes that were put into this sweet corn that coded for some different proteins, similar but different. It also had a BT gene for rootworm, so it protected against that root feeding beetle larva. And it had a little bit of tolerance to Roundup Ready. So that's what we're talking about with this one. How has that performed? Again, this came in around 2010 or so. And first I want to say these slides were from Galen Dively, who put them together for his talk. And, these, it summarizes all our data though. You see the number of trials that are there. So we were getting about 80% control when this thing first came out, this performance series BT sweet corn, and currently it's really plummeted as well. Only about 17% reduction in ear damage with our current trials. What's happened here? Same thing. Corn earworms develop resistance to the BT. So it's horrible. It's horrible if you were depending on BT sweet corn to take care of all your pest problems, because with any of these, it's not anymore. So fortunately, we do have some more genes that have been put into sweet corn, and the attribute 2 or attribute plus, it's called, from Syngenta. And these are, this BT has a gene called the VIP3A gene, in addition to the other Cry1AB genes that we talked about. The addition of the VIP3A gene codes for a protein that is currently very toxic to corn earworm. So it works, in other words, and that you can see the different corn varieties that are out there with this one. And I'll show you the data. We're really getting about 99% control of corn earworm. It's a good way to summarize it with this VIP3A. And it's very rare that you see a damaged ear. And sometimes we're wondering, when we do get that damaged ear, what's going on here? Is there some reason that one plant didn't express it? So that's good news. The bad news is that's it. There's nothing after that. Although we'll hear in our next talk whether there might be, but we're really riding on one gene currently that still works for pretty much complete control of corn earworm with BT corn. So here's the typical trial you'll get. There's only one type of variety, like Remedy in this case, that has the VIP gene that's going to give you, excellent control, no damaged ears, and you see all the rest of them kind of given, working about as if you had non-BT corn. So that's kind of it for BT corn if you're using that alone. Now I will talk about how you might use and how it might be beneficial to actually use a little bit of insecticide application along with the BT sweet corn. I know you're wondering, well, heck, I paid premium for getting this BT gene, and now you're telling me I got to spray. Well, the one thing we do know is the performance series BT corn, we'll get damaged ears, but the one thing we don't get is large larvae. The larvae don't develop, they stay small, they're sick, they're a little bit poisoned, but they didn't die. So it counts as a damaged ear, an infested ear, but it's not damaged too bad. And the larvae doesn't get too large, which means it's not doing a lot of feeding. It's also very, very susceptible to insecticides. So if it's weakened by the BT corn, it's a lot more easier to kill than it's a lot slower moving. So maybe the 1-2 punch could be some of these other BT corns that might not be working, but then a little bit of insecticide complementing them. So that's a strategy that you might consider. But now we're getting into bridging into insecticides and some of the work that we've done too. So if you're a conventional grower, and I will say I don't always know the regulations in New York and Long Island and what can be and can't be, but I believe Coragen and Vantacor, the group 28s aren't allowed for use on Long Island. Someone can correct me if I'm wrong on that one, but we'll talk about the options that most growers have, conventional growers. You're really looking at an older carbamate, lanate, that still provides good control of corn earworm. It's out there, a different class of chemistry. A lot of pyrethroid use. You see them group 3. I've listed many of the products, but not all of them. There's plenty out there. You have spinosins, which include products like Blackhawk and Radiant. These are a completely different mode of action, very effective on Lepidopteran larvae like corn earworm. You have diamides, such as Coragen, and you may not recognize the word Vantacor. I bet you if I ask everyone to raise their hand, have you heard of that? Probably no one has, because it's brand new. What it is Coragen, about twice as concentrated. So it's a new product that's going to be available, and it's basically going to fit the same niche as Coragen, which means it's labeled on sweet corn. And I can't talk about price because I'm not selling these things, but when it's twice as concentrated and now you've got 2 products, that usually means good things for a reduced price. So that's going on. You've got combination products that have, like Besiege, that have both the pyrethroid and the Coragen in a combo product. And now Elevest, another one you may not have heard of, is going to be offering kind of the same package, but it's a different pyrethroid. So Elevest has Coragen in it, plus the pyrethroid bifenthrin. So it's a brand new product, again, kind of same niche as Besiege, just another product that you have. And usually the more products, the better for the growers, as you're talking about competition, and that's a good thing, and more offerings and more options. So I'm not going to talk much about the biologicals on the bottom because I'm going to end my talk talking about organic sweet corn, and we'll talk about those then. So moving on to insecticides again. So pyrethroids have been historically, for the last 20 years, the #1 used product in sweet corn. They've worked brilliantly against corn earworm for decades, and there's a lot of products, a lot have gone generic, and a lot of different options, and low cost. It's a big reason why they've been used, and they've been effective. Broad spectrum. You might recognize these, you might use these, a lot of these still. Problem is, around 2008 or so, what you're looking at on this chart is corn earworm vial tests, where they basically catch the moths and put them in treated vials with a pyrethroid and treat it at a point where you should kill all the moths that go in these vials. And when you don't, it's an alerting that you're getting survival, and that survival means they're carrying genes that are resistant to pyrethroid. So we started picking up in Virginia, these are some data, that all of a sudden in 2008, about 20% of those moths survived, and that's stayed steady over the years, if not gotten larger. So we had big time resistance pop up since 2008, and it's in the populations, and it's in corn earworm populations across the country. So that was the alert. But then from the standpoint of the growers, what does this mean? And it took us a long time to see field level failures where growers were spraying and we're just like, you're not killing these corn earworms at all with pyrethroids. But then we started about a couple of years ago, we would put some trials out, and we sprayed pyrethroids, which used to kill 90%, and now we're only killing about 50%. So that led us to a project where we looked at in 2020. Is that across all pyrethroids, or is this just something that might be going on with some of them? Are any pyrethroids better than others? So this is what we looked at. We had 2 locations in Virginia, and you see here is a list of a bunch of pyrethroids, the most popular ones. And keep in mind, on the bottom, treatment 10 is Besiege, which is both a pyrethroid plus Coragen. That's the only one. The rest are all pyrethroids. We tested this in 2 locations, really good coverage. We basically doused the ears and the silks with sprays on a single nozzle boom and put on 7 applications. So really good coverage and a lot of sprays. So it's really up to the insecticide. Is it working or not? Well, one of the locations, Whitethorn, we actually got great control with everything. So all those pyrethroids, they're all working. They're all working pretty well, very comparable to Besiege. And there you go. So that was one site in Western Virginia. And that kind of surprised us. We actually thought we should see a little less control with the pyrethroids. But then we look at the Eastern Shore, and there you see the drop off. We're only getting about half the control we used to, and that was consistent across all the pyrethroids. Maybe Hero was working a little bit better. Hero, by the way, has 2 pyrethroids in it. I don't know if two modes of action, 2 pyrethroids are better than 1. I often doubted that, but now I'm starting to wonder. But then you can see how Besiege performed, and just really the only way you're getting excellent control is if with the addition of the diamide. So pretty clear we got resistance going on the Eastern Shore. What we don't know, and what you may not know, is farm to farm, what is your corn earworm resistance levels? And that can vary, as you can see from the data from 2 Virginia sites. Other thing you need to keep in mind. So we're talking about reasons that maybe going with a straight pyrethroid rotation might not be the best thing anymore. And a lot of growers you were spraying pyrethroids every spray. 1, resistance problems. 2, what else are you killing when you spray pyrethroids? Something that maybe you don't think about. But there are a lot of predators that come to sweet corn, especially when it starts silking and producing pollen. Things like lady beetles, little tiny predatory bugs. I've been out there in these fields counting these things for research studies. They're out there and they're doing a lot of things. And one thing they do is eat corn earworm eggs, and they eat tiny corn earworm larvae, among other things. So when you spray pyrethroids, you kill these. They are very, very susceptible to pyrethroid sprays. So you're spraying them, and you always have, and you need to keep corn earworm out. But man, keep in mind you're killing something that's on your side that they're doing a lot. So can you spray something that might not kill these guys and these girls? And yes, you can. There's going to be some other insecticides such as Coragen. But I do want to say I've seen this in Virginia. I'm guessing it happens in New York too. You spray pyrethroids a lot, you might end up getting an outbreak of this. This is corn leaf aphid. This is an aphid that is resistant to pyrethroids. And when you kill all the lady beetles and the things that eat the aphids, they just explode on you. And so bad sometimes they cover the husk. You get sticky honeydew from the aphids and sooty mold growing on that. And it's not a good thing if you're trying to sell that corn. So a problem brought on purely by spraying pyrethroids. So moving on to some other chemistries. So maybe you've kind of gotten in your head now, it makes sense to rotate some other chemistries in there. The Coragen and Vanacore are a good option to bring in, as are the spinosins such as Blackhawk, Radiant, Intrepid Edge, for a lot of reasons. One is they're more IPM friendly. They don't take out these natural enemies. So they allow them to do what they're doing. And then you're also not selecting for resistant corn earworms for pyrethroid resistance. So the question is, how well are these things working? We've run trials for the last, it's probably been 10 years with Coragen, where we have looked at straight Coragen for all sprays. And then that doesn't really make a lot of sense because you will max out after about 3 sprays. You can't put any more according to the label. Same thing with like a Blackhawk or a Radiant. So you're always going to probably rotate these with other chemistries. So what we looked at in recent trials, actually since 2015 or so, is rotating these in with a pyrethroid. That's something that fits what a grower would be doing. So this is what you're looking at here, is just a trial where every other spray was Hero, and then the other sprays were what's listed here. And you're getting decent control. This one, I don't know why I didn't put the percent damaged ears, but you can see the number of larvae that were in the ears. And Coragen, Besiege, Radiant, all doing pretty well in a rotation. Maybe a drop off with Blackhawk. Here's
So
a trial.
Tom, hey
Yep.
Tom,
Yes.
so 7 sprays. So as you said, so on the last one, so or 4 sprays, but there
Right.
was
And
no all. You alternated every other week with a pyrethroid on this trial, or this was straight?
yeah, and thanks for pointing that out, Chuck. This one, so this is in the mountains of Virginia, and we wanted to see if we could get by with 4 sprays only. This was kind of a trial. I didn't think we were going to have much corn earworm pressure, so we tried at 4 sprays, and you can see the spray dates there. They're basically 7 days apart or so. I think we needed more sprays overall, but that was that one. This one you're looking at 7 sprays, so this is probably more what we're doing in the eastern part of Virginia and maybe same in New York. I don't know if you put that many sprays on, but same thing here though. We rotated with Warrior every other spray, and you can kind of see the control here. And what I want to point out is Coragen and Besiege are good options for this rotation. The big difference between Besiege and Coragen is Besiege is bringing the pyrethroid with it. And so you're not doing anything for resistance management. You're not doing anything for not killing natural enemies. You're going to do that with Besiege. But Besiege has a diamide, which works pretty well on corn earworm, as you can see. So see, also shows maybe the little bit of a drop off with Blackhawk, the spinosyns. Going on in this trial, and that's pretty consistent against all our trials. Another thing that you might consider is, and you've probably seen this, and I know I have, when corn is pollinating, sweet corn in particular, the bees come. Man, we see it just buzzing with bees. And corn obviously is a wind pollinated plant, but when it's producing pollen, the bees are coming and grabbing that up. And we don't want to kill them. And spraying Coragen when corn is pollinating seems to make a lot of sense because that's pretty safe for the bees. It's probably the only one in there that has zero or low toxicity to the bees. So maybe a good time to fit Coragen into your rotation is when the corn's pollinating. That's kind of what I'm saying with that one. Some other considerations, and again, I'll thank Galen Dively for these slides. He and I, for years, have shared slides and projects we collaborated on, and whoever has the best slides we end up using for these. So this is a Galen Dively slide. Good things to keep in mind. So when you're spraying sweet corn, the goal is to protect that ear zone. That's where all the action's happening. That's where eggs are being laid on silks and they're going right into that ear. So that's really the only thing you're worried about protecting anymore when corn starts to develop the ear. So whatever you can do to target that spray, hitting the ear, hitting the silks is going to help you. Going with aerial spray, and sometimes you got to do what you got to do. But keep in mind when you're putting something out like an air blast or an aerial spray that you might not get the coverage that you might get with maybe a high boy that's coming over top and directing the nozzles right at the ear. You've got to do with what equipment you have and the size of your corn and all that and the drive rows you have. I get all that, but no, the level of control you're going to get is really dependent on how well you can get the insecticide to where it needs to go. Other thing to keep in mind, silks grow over a period of maybe 4 to 6 days. You've got silks coming out of that ear. And whenever you get additional silk growth after you spray, that's unprotected tissue. So if you spray and all of a sudden you have maybe 4 days since you spray again, and you get a lot of silks growing, you probably got a lot of susceptible sweet corn out there. A moth can come flying in, lay an egg on a silk that never got touched by an insecticide. That egg hatches, larvae crawls right down and gets in the ear. So understanding that can help you with knowing that you've put a good spray on because it was, the silks were out there and you hit it well. All right, moving on to another question that we get a lot, which is sap beetles. So, we've sprayed pyrethroids and they've always taken care of sap beetles. So now you're saying spray something else. And well, this is what kills sap beetles. Pyrethroids do, lanate does, besiege does. and the spinosins like Blackhawk Radiant do. Coragen does not. But we do know that if you control corn earworm really well, you're pretty much taking care of the sap beetle problem. That 80 to 90% of the sap beetles will get into a sweet corn ear behind a corn earworm. So they basically use the entry hole that a corn earworm has made to get into the sweet corn ear. So if you control corn earworm, you're going to take care of most of your sap beetle problems anyway. So it doesn't matter what controls them. All right, I'm going to move into sweet corn. Assuming we have some organic sweet corn growers, I'd say raise your hand, but I can't really see all the people anyway. But I'm going to assume that we may have some organic sweet corn, or maybe even some growers that are interested in getting into it. So I have given talks every year at meetings where they focus on organic agriculture and biological farming, and I have never seen so much advertisement for products and things that'll do this and things that'll do that, natural products and when you're talking sweet corn control of corn earworm, that is one of the hardest pests and crop situations that we have. It is really, really difficult to control that pest. So to say you're going to do it with pepper spray or some, it's like, good luck, because this is one tough insect to control. But there are some products that do have activity against corn earworm, and we've looked at them through the years. Pyganic and Trust, the BTs, to name a few. And I'm gonna get into some of these and possible options here for organic production. Okay, the BT products, you've got like Dipel, Zentari, Agree. I'm trying to think of such Javelin. These are all types of BT sprayable products. One of the problems, do you remember when I was talking about the BT transgenic corn? What happened to that first event that came into the 90s? It used to work, remember? And then now it doesn't work at all. That happens to be the same protein that's in these products. So what do you think? Doesn't stand a chance now, right? And that's exactly what's happened to these BT sprayable products. They worked once before, and they're not working anymore. Corn earworms develop resistance to that BT protein. So just to show you, we tested it last year in a trial, and here's just an example. 2 different BTs sprayed 7 times, and we basically got the same damage that we did in the untreated controls. So BTs aren't going to work. How about some of the other products that an organic grower might use? Things like Nemix, which has azadirachtin in it, Pyganic, which has pyrethrins, natural products, and Trust, which is basically the same as Blackhawk. So that's spinosin. And we know that's going to give you a little bit of control. And here's a trial by Galen Dively, University of Maryland. It was a really good trial. That's why I'm showing it. And he got no control with the neem product, no control with Pyganic, and very, very good control with Entrust, which has spinosin, as you can see in the red there, percent damaged ears. So there's no doubt the best option in organic sweet corn control is Entrust. Problem is you're limited on the number of applications, or you'll max out. So you're going to have to use those sprays wisely and probably rotate to something else. Okay, some factors with organic control, but really any control, is to remember about those silks growing when you're putting these sprays. If you only have one really good product like Entrust, timing it is going to be really important, and hitting it when those silks have really come out, but not being too late. Monitoring for moths to know that you have a lot of activity now is some pretty important things. Another thing to consider is that products like pyrethrins and spinosin that's in Entrust, they break down pretty quickly in sunlight. So although they might work, they're not going to stick around. You're not going to have a long residual with them. And you might need a nice 1-2, every 2 to 3 days to really get good results. All right, here's a product called a Zeolater. It came out of the New England. Ruth Hazard and some others developed it, and It's basically a way of dispensing some kind of oil, often vegetable oil, onto the silks, and that carries with it maybe an insecticide. Used to be BT they used, but now I think you should use Entrust. And it basically it makes sure that that worm is going to ingest the insecticide where it's going to work well. It's very targeted. And from an insect control standpoint, if you do it right, you will control your cornea worms. Um, so, but the problem is just take a look at that. I don't know how many people are familiar with it, but it's pretty labor intensive and time consuming. You're going around individually to every ear, applying an oil, dispensing an oil on the silks. So maybe not something for large acreages. It's also been a lot of reports of some issues with pollination and then kernels developing because of the oil you put down that interfered with pollination. So there's some issues and some problems with that technique, and also when to do it. We've talked about the silks growing, and you don't want to be doing this zeolater multiple times. That's a lot of labor. So you're looking at something that hopefully you were going to do once, if at all. And now you're talking when to do it is going to be such an important time. You could do it too late or too early. So it's a tough strategy to really get down well. And it's definitely something for more smaller growers that can pull this off. But one more thing for organic growers I'm going to talk about, which is a product that's been around, but now they've got a newer product called Heligen. The Gemstar's been around a while. What this is a virus particles that are very, very specific to cornea worm. All they kill is cornea worms. It's a specific virus. And basically you spray the virus particles out on your field, and the cornea worms ingest them, and they eventually die of this virus disease. It's OMRI certified, doesn't kill anything but cornea worms, so IPM friendly, bee friendly. How well does it work? We have gotten the best results with these products like Heligen and Gemstar in hemp. We're doing hemp insecticide trials, and cornea worm's the #1 pest of that crop. And what you see here are the diseased larvae after spraying these products. So we spray it and we see the diseased larvae, and we know it's working. It's just, is it working good enough? And that's the big question. And sweet corn, it's a really big challenge. We got promising results from an actual on-farm trial we did in Blacksburg, Virginia, Wall Brothers Farms, where we used an air blast sprayer, put out Heligen twice a week from tassel push to harvest. And you see 2 different rates. We're spraying this virus particles, and we actually got an increase, significant increase in the percent marketable ears compared to not spraying at all. So, it was, now I will say a marketable ear had a little bit of tip damage. So this isn't clean ears. This is marketable ears, assuming that a little bit of tip damage was still marketable. But on the large scale, on the 1/3 acre plots, we actually got something that was a little promising. Problem is, when you look at Heligen in trials, small plot trials, like replicated, randomized, like this one here, it ends up paling in the comparison to the other treatments. Here's a great example. Get the Heligen sprayed for all the sprays, 7 applications, compared to spray #2 is kind of, I call it the Cadillac. This is the best grower spray program that a grower would suggest, and that's a rotation of chemistries. You got pyrethroids with lanate and coragen, and then we've got straight warrior, and then Heligen rotated with warrior. And there's your percent clean ears. The only thing that gave you any hope was that Cadillac treatment. This was pretty heavy pressure. Heligen not really knocking out the clean ears, but most of the damaged ears with Heligen, it's in the tip. It's just a little bit of tip damage only, but still a damaged ear. So I'm not writing it off. I think there's something there. I think we got to find what to rotate that with, but getting some virus particles out early in the field is a good thing, especially if the climate fits well. So that's all I've got. I want to thank all the students that have worked on these vegetable plots, and Helene Dowdy on the Eastern Shore AREC, who puts out trials out there for our program. And I don't know if I have any time, if I've left time, Chuck, for any questions, but I'm happy to answer any.
Yeah, actually, Tom, I think we've got a couple questions that came in over the chat.
Sure.
One of them was uh, on the trial where you were comparing the pyrethroids on the two farms. Were those sprays on each farm, were they the same in terms of did you use same adjuvants? Did you use the same gallonage of water? Did you use a similar sprayer? Um, that was one of the questions that that popped up about that study.
Yeah, very, very good. We didn't have any adjuvants, so these are just straight pyrethroids mixed with tap water. We spray the same way in both. I mean, it wasn't the same sprayer, but it was a CO2 backpack sprayer with a single nozzle boom, and we put out 30 gallons, 30 gallons per acre. Now I think it was 20 to 30, somewhere in there. At both locations. So yeah, we sprayed the same way, really targeting the ear. So I don't think it was anything in the spray. It was really the location and the resistance to pyrethroids that varied across our two farms. We're pretty convinced of that, but very good question.
Okay, another question that popped up was, do the BT sprays still work on earworm, and should you worry about cross resistance? And you kind of touched on that, but I'll it was a question, and I just want to just make sure you re-emphasize what you said.
Yes, I would stand by the fact that BTs don't work for corn earworm anymore, and at least the ones that we've tested in Virginia. And we've even gone as far in hemp, because that crop's got hardly any insecticides, and it does have BTs, and corn earworm is killing it, and the BTs aren't working. And we have actually dunked seed heads of hemp in the BT solutions and put corn earworms in diet cups, and we're not killing them. So it's resistance. Resistance has developed to the BT transgenic corn, and it's caused the BT sprays to not work anymore. Is what's happened.
Okay, another question was, would adding a surfactant or adjuvant help with coverage later in the season?
Great question. And I mean, I'm going to say I don't know, because I think there's a whole area of research that needs to be done with what types of adjuvants, and whether they work. I've sat in on studies, and I've seen where this adjuvant will make all the difference in the world, and then you talk to someone else and it's like, yeah, that's just snake oil. And I've not done the studies myself. I'm so busy testing insecticide chemistries that factoring in the different adjuvants ends up another layer of things. But I will say I've gotten more and more interested, because growers have asked that question, just what happened. And I think it's some studies that we're going to be doing. We have a group of entomologists in the Mid-Atlantic, and even some in New York, who are going to be collaborating on some things that we're going to do to help the sweet corn industry battle corn earworm primarily. And one of the things that we're probably going to look at is there any benefit to some of these adjuvants? Yeah, increasing residual activity, it would be the best thing that it would do. Maybe help, stick it on that silk, and that might improve efficacy of insecticides too. So great question. I can't answer it because I don't have my own data yet.
Well, we'll hold you to it then. At some point in time, maybe you will be able to answer that.
Yeah, maybe a talk next year or 2 years from now if we do it.
So. Another question, and Jay stole my question from that I had, but we both wanted to know: have you looked at Spearlep?
Yeah,
that's the BT.
yes.
That's with the peptide.
Yeah, Spearlep. We have looked at it. So for those who aren't familiar, this is like a spider venom peptide type of toxin. That's how it kills the insect. And it's very interesting and very exciting, because it will kill the insect. It's just, how do you get it in the system of the insect? And there's two strategies that they're using with this new insecticide. One is, if you use it with like a BT, what the BT does is opens up a pore in the gut, in the midgut of the insect. And how the BT would work on its own is it opens that pore up and the gut content spills out and the insect dies of septicemia. But if the BT opens up a pore and now all of a sudden this toxin can get in the system of the insect, it's going to kill it. So that's kind of what they're looking at, is that 1-2 punch. We didn't get very good results in our trials last year. It was our first year looking at it. I think we need to hone in on some things with the combination, and also the fact that our corn earworms have developed resistance to BT may have complicated that whole strategy. So that strategy maybe on some other insects is going to be more promising. But however, we have looked at, kind of off topic a little bit, that same insecticide, Spear Lep, is going to be a product called Spear T. Same insecticide, just marketed a little different. You can put it out at higher rates. If you basically drowned an insect in it, spray it where you're basically getting it in the spiracles, which are the pores that the insect breathes through. If it can get in there, it's actually toxic. And we've killed stink bugs with that other product in some trials, and squash bugs and things like that. So there still needs a lot more science done on it. It's great that someone brought it up. It's kind of a new exciting product, and one that I think we need to work at how this is going to actually work well with our different pest situations. But we didn't get very good control with combining it with BT last year, but we do have BT resistance, so that might be a problem.
Okay, and then I think last one for right now, and that is, if you spray at night, are the honeybees safe?
Yes, that's another way of reducing your pollinator impact, is, wait for those bees to go back, if it's honeybees, back in their hive. And so yeah, you can definitely help the situation by not spraying when the bees are active. So yeah, that's a good point. And honeybees, and this isn't brought up as much. So there's a lot of toxicity testing done on bees and showing that it hurts them there, and it could hurt colony, it hurts their behavior. But what you don't bring in is, are bees repelled by the insecticide? And if they are, they're not going to come into the field. And that's kind of a whole new aspect that needs to be looked at, because honeybees know when a pyrethroid sprayed and they stay away from it. And so there could be a little of both. You're spraying when they're not as active, and you're also spraying something that they know to stay away from anyway. So I'm kind of getting off topic with this, but I think with pyrethroids, you're probably a lot safer than we used to think, but it is a good idea to spray when they're not in their peak activity period. And yeah, getting into the evening and night definitely is much safer.
Great, thanks, Tom. Hopefully you're going to stick around for a little bit, because I know I had one other question, but I was going to hold off until later on.
Yeah, sure. Yeah, thanks.
Okay.
Thanks for all the questions.
All right, go ahead and stop your share. And Chelsea, if you can launch the first poll, that would be fantastic. So again, if you get a chance, if you can just answer the poll that's on your screen. I knew somebody would select neighbors. All right, great. We're about halfway. All right. So it's a pretty close. Looks like birds come in for the worst pest on their farm at 41%, followed right behind by insects at 32%. Neighbors at 5%, and weeds at 22%. So thank you very much for sharing in that poll. I appreciate it. All right, and then go ahead. We'll get rid of that poll. So I'm going to introduce our next speaker. So going to kind of continue along this theme. I know many of you know our next speaker. So I asked Blake Myers, who is now with Stoke Seeds, to talk about where are we heading with sweet corn genetics. Blake was currently with Seeger Seeds and is now a seed consultant for Stokes. His territory includes Pennsylvania, New York, and New England. He joined Seegers in 1994 after working for Syngenta and 11 years at Harris Moran. He has his bachelor's degree from Purdue University, and he specializes in sweet corn and evaluates between 500 and 2,000 varieties per year. Blake will discuss genetically modified sweet corn and their impact on the control of sweet corn pests, and then just kind of where we are heading generally with sweet corn genetics. And I know a lot of the transgenic materials, it poses a lot of questions for our growers and whether or not to use them for marketing. So with that, I'm going to ask Blake to go ahead and start sharing his screen.
Okay, Chuck, can you hear me okay?
I can hear you. Yep.
Now I just have to figure out how to share my screen.
Well, we're halfway there.
You guys got to forgive me. I'm kind of a newbie at this. Okay.
But you nailed it earlier, so I know you can do it.
I know I nailed it earlier, but that was a while ago. Okay, let me escape out of this. And
the pressure's on now, Blake. That's all.
What's that?
The pressure's on now. That's all.
I know, Chuck. Told you should have found somebody with more experience. Okay, so where do I turn this on, the screen sharing at?
Should be down on the bottom. Little menu bar should pop up. It's a green button, says share screen.
There we go. It popped up. Sorry, I had something else on there. Okay, are we seeing it now?
Yep, we've got your desktop.
Okay, and now we'll get to the slide presentation.
Yep, there you go. Okay.
All right, guys.
Thank you, Blake. Take it away.
Yep. All right. So like Chuck said, I'm going to give you a bit of an overview about where we're headed. And Tom, you did an excellent job. You helped me out quite a bit, actually, in being a scientist. And you put a nice emphasis on what we got to concentrate on. So I used to, like Chuck said, I went to Purdue and I had a professor there that always started out with, well, why are we all here? And he'd answer, because we're not all there. But today's tech, today has put a different spin on things. Today's reality is we're not here because we are all there. And why are we all there? Well, obviously, because we're not all here, and we got to thank Zoom and WebEx for that. So it makes things incredibly different than what it used to be. I can't look out in the audience and see your reaction, so just kind of bear with me. But the real answer is, we're here because of technology. Okay. And I think that I can keep my talk fairly simple. Okay. If Einstein can, basically describe the universe with E equals mc squared, I think I should be able to handle this in 30 minutes. In fact, maybe even less. It comes down to basically this. If it's not attribute plus or attribute 2, you'd better spray. Thank you. And that's the end. No, just kidding. A wish. But anyways, so we're going to look at why we modify organisms, what were some of the traditional breeding organisms, and what species are GMO. I like the term genetically engineered better than GMO, because as you all know, everything's pretty much modified, but not everything's engineered. What are the concerns and so forth. So anyways, we'll jump into that. So let's look at the timeline along here, because I want to make a point about how fast this is progressing. Okay, so you got to ask yourself, you wouldn't use the same cell phone that you used back in the 1980s. We're all pretty familiar with the Motorola brick phone, or, those of us that are old enough. And it cost about $4,000, took 10 hours to charge, and you had a 30-minute talk time. Or the same computer that we used back then. This computer is far less powerful than probably your cell phone. It had a $500,000 annual lease. So anyways, things are advancing. And, I live in Rochester, New York, home of Kodak. And even Kodak didn't see this coming. In the 1990s, who would have ever thought that you'd never use film again? So the thing is, when you look back, things that seemed foreign to us just 10 years ago are commonplace today. And they're not all welcome by some people. Okay, so your perspective on things, and here just to emphasize the drone shot, can give you a different look at the world around you. And in here you can see, the problems in the field and so forth. Okay, so along came the BTs. And I think everybody kind of expected a silver bullet. We didn't have a silver bullet here because, as Tom mentioned, we had the cry event, BT11, and it wasn't all that. It started out being fairly effective, but it built up resistance relatively quickly. Okay, so let's take a look at how we got here. Up to 7,000 years ago, I'm sorry, let me go back. We started out with something called teosinte corn, and it really was more of a grain crop than what it was a sweet corn or a field corn. Then, I found this book from the 1930s from Cornell, and it shows, your various corns that were popular in the early 30s. These were, of course, all open pollinated. They took a lot of years to select, maybe 15 years to select a single variety and move forward. You'd select them for things like earliness and row count. And here, Mr. Whipple selected it for color and again, earliness. And then he went on to do the Charmin ads. I'm just kidding, of course. But color was part of it. One of the things that really amazes me is the low row counts on these corn varieties back then. The color, like this black Mexican, I can't imagine that would look too appetizing boiling in a bowl of water. Mammoth, or then you have the midget corn here, like you had with maybe Mirai minis. Okay, but the first widely grown sweet corn hybrid was Golden Bantam back in the 1930s. And this opened up a new era for, breeding corn. This is a picture of inbred corn lines. And as you can see, you inbreed a corn for about 8 or 10 generations. Well, that's probably not right. 7 or 8 generations, we'll say. And you end up with these inbred parents that are small and weak, but they're genetically very uniform. And you can cross one line with another. So if you come up with a female line, we'll say, and a male line, you can cross them with each other and start to develop hybrids relatively fast, I'd say 3 to 10 years. So we start with all these theoretical crosses on a computer. Then the breeder makes these thousands and thousands of hand crosses, maybe 10,000 a year. And then it comes down to hundreds of local short row trials and dozens of local block trials. And there's this giant filtering out process that takes place. So this is one of the variety test plots we planted one year. There was me and this other guy planted 2,000 varieties that year. And it's not always the best conditions. You do it with, the state-of-the-art equipment here, the hand stabbers. And sometimes this is an actual field with those rocks in it. And there's our packet. You had tried to get in there. Then you have to go back and evaluate each one of these too, and I take 40 to 50 data points on them. So again, going back to this time frame thing, in 1998, just 23 years ago, Rogers offered its first traded sweet corn variety, which is an attribute 1 variety, and at the same time, Monsanto introduced its first Roundup Ready corn. So about 10 years ago, and let's look back now just 10 years ago from today. 9/11 was already 10 years behind us, and you were probably growing things like BC 0805, Garrison 7143, 1170, Devotion. You folks are familiar with those. Okay, so let's look at some of the row crops that were GMO. And again, this, because not everybody's pleased with the fact that things are GMO, I want to show you a little bit of maybe the concern is a little overblown on some of this. You tend to think when you look in a seed catalog that everything in that catalog must be GMO, or certainly everything with a number. Well, that's not correct. So, of course, we have the sweet corn for the herbicide tolerance and the worm control, and some summer squash, but only for virus. When it comes down to it, there's only about 10.8% of the sweet corn are genetically modified that are sold. Okay, and in the summer squash, it was even less than that. It was only 1.5. Okay, so again, putting this in perspective, this is the field corn acres in the United States. They make up 98% of the corn in the United States. Here's the sweet corn that makes up that other 2% if you were to stack it on top there. Now, looking over here, genetically engineered corn is about, roughly 10% of that 2%. That line is here, but you can't even see it. So looking down from here to this, the genetically engineered sweet corn is, well, we consider it important, but maybe the breeding companies and stuff don't. Okay, so look at the trends here. So when you look at, the years from 2002 to 2017, okay, this is the percent corn that was conventional, this blue line, and this is the percent corn that was GMO, sweet corn. This is based on sales. I think I looked, something like 30,000 purchases of sweet corn to make this data. So, I mean, it's based on the Northeast, but I think it's pretty reflective of it. Your roadside growers then tended to use a little bit more than your large commercial growers. So, again, here's your trend lines, what percent roadside your user was using. And this is the genetically engineered corn down along the bottom here. See it, I don't know if it's an anomaly, but it's starting to rise up a little bit here toward the end. Okay, so then your small growers, these are even smaller growers. I would say either a tiny roadside grower or a big gardener, they're up to even a little above 20% of what they're using. But the thing is let's go back to your large growers. I mean, your large growers are not using very much. Medium growers a little more, small growers a little more still. So that presents a problem because, let's see, I'll jump a slide or two ahead. Okay, if you're spending this kind of money to develop a trait up to $130 million, you're not breeding it for sweet corn. So what we're doing, again, back up here a little bit, you're using the sweet corn as a platform for the delivery of those traits. But when a Monsanto or a Bayer or a Syngenta breeds, they're doing that for a field corn, then they're back crossing the trait into the sweet corn. And so they're just using the sweet corn, for lack of a better word, almost as an additional sales, but they're not going to risk the sales of all those row crops for sweet corn. Okay, so here's an example of a successful sweet corn variety that is used as a platform. So I don't know, probably in the early to mid 90s, Serendipity was bred by Rogers NK, which later became Syngenta. And then they added rust resistance, that became Providence. They added BT11, the cry gene, the BT event 11, and that became BC0805. And then they added the VIP gene along with the BT genes, that became Remedy. And along with Remedy, you got a few tip issues, so they straightened those out, and that became Pursuit, which we're using today. So this is using the Serendipity as a chassis and moving forward. Here's another good example of it and how this can work. So you take a company like Illinois Foundation that does not breed genetically engineered crops at all, and yet they supplied Anthem to, I think at the time it was Monsanto, or Seminis, and they added the BT gene to it, and it became Anthem XR2. So now you've got Anthem with the performance package. Okay, so let's look at the average sale of a genetically engineered sweet corn. The average sale that comes into us is about 88.16 thousand seeds, so it's less than 4 acres. The median sale, when you look at it, is about an acre's worth at a time. Okay, we already looked at this slide, but again, this is why they're breeding for field corn. So I guess my take-home message on this is that these genes were not bred for sweet corn. They're back-crossed into sweet corn from field corn. Companies are not willing to risk their field crop business for sweet corn. And some companies will license their genetics to be used as these platforms, and then there's cooperative breeding between these two companies. Okay, the genes that are involved, Tom went over this pretty well, but there's the cry genes, which are for the crystalline proteins. And as he also showed, that resistance was built up fast to it, and they're not very effective anymore. Then there's the VIP Ptera genes that are used for, and that's the vegetative insecticidal proteins. VIP3A is the proprietary is Syngenta, and both came from BT bacteria, the Bacillus thuringiensis. So I think the important thing that we want to look at on this chart is that, you know, again, if you're looking at something that's effective for European corn borer, corn armyworm, fall armyworm, black cutworm, but also I would say Western bean cutworm, as long as you've got this, as long as you've got this VIP gene in here, you're doing well. And, you know, in addition to the cry gene. If you're going back to the old attribute varieties, you're not getting very good control of anything but European corn borer. And, you know, it's effective against them, but I think it's also, Tom, I think it's effective because it has to feed so far up the stalk to get into the ear also. It's consuming a lot. Then you have the Seminus Performance series with only the 2 cry genes. And yes, again, it's effective against the European corn borer. Corn earworm, I mean, I would not say very good, but that's, you know, I guess that's a matter of opinion. And basically what I'm noticing with this performance series is you have to spray as much and as often as you do with a non-GE corn. So here's your non-BTs, here's your attribute, you know, attribute 1s and your attribute 2s, and we're not seeing any significant breakdown at all with the attribute 2s in a timeline. Okay, what are the industry positions on breeding the genetic engineered traits? Seminus, yes, I'd say they're definitely into it. They are planning to release a couple new varieties in the next probably 2 or 3 years. They will only have the performance series package, so it won't give you ideal coverage. They're using a couple of their varieties as that platform, that chassis that we were talking about, which would be 0313 is one of the new varieties that they're coming out with. They'll use that. Syngenta is definitely moving ahead. And again, they're trying to stay with the attribute 2 series. Okay, now you might want to ask, what is the attribute plus series? It's somewhere in between attribute 1 and attribute 2. Attribute 2 was bred to have Roundup resistance, glyphosate resistance or tolerance, but that gene turned out to be a little bit tricky and it didn't always express itself. So on a variety like Patriarch, even though it has the genes in it to be glyphosate tolerant or resistant, it's not expressing itself. So they don't call it an attribute 2. They had to come up with something sort of in between. You can spray Liberty on it, but not glyphosate. Okay, Illinois Foundation, again, they're not going to be breeding for any genetically engineered varieties, but again, just like they did with Anthem and Anthem 2XR, they're going to allow their genetics to be used as platforms. Crookham has no intentions of going with any breeding, although again, they may allow some of theirs to be used as platforms. Seneca Vegetable Research, no, and Harris Moran, they're owned by a European company in France, and that's a definite no. Okay, so this is sort of just a lineup of the varieties that have been out there and what trait they carry. I would say this is not entirely inclusive, but it's a pretty complete list. Okay, so these are the ones that are showing to date really good worm resistance. Okay, and there's not a lot of them. Quick Trip, which is basically Alto, that's an SE, an early SE. Pursuit, which Tom mentioned, and that's working very well. It's selling very well, but it's exceedingly short crop this year, and we expect Pursuit and Remedy to be short next year also in supply. In Aspire, you don't hear too much about because that's a yellow version, basically the yellow conversion of these. Then you get into, you know, the Patriarch and the Milky Way, and Patriarch in particular is lacking the glyphosate tolerance. It does have the two BT events, the cry and also the VIP genes in it. So it's giving you good worm control, but you can't spray Roundup on it. Milky Way is sort of even in between those. Most people are spraying Roundup on Milky Way and they're ending up with 6 to maybe 15% plant death with it, but they feel that it's worth it to go ahead and spray. And then we get down in, then we get down into the performance series. And again, you know, you're buying this mostly, I would say, for the glyphosate package and control of the European corn borer. Okay, so this is basically what Illinois Foundation is telling us about breeding new ones. All hybrid sweet corn produced by Illinois Foundation has been developed by using traditional plant breeding techniques. These varieties were not developed using genetic transformation or other types of genetic manipulation. It is possible that Illinois Foundation will provide the genetic base or the foundation for a hybrid, but it will not be making conversions of those hybrids to GMO. You know, so it's basically going to supply their platform to other companies if they can strike up cooperative breeding or releases. Okay, I'm not going to go over a lot of resistance. Tom did a great job with that, but you know, I think it's enough to say that the VIP traits are holding up well. The timing of the sprays, again, just to reiterate what Tom said there, is you've got to get it on early when the silk is first coming out. Okay, so, you know, that was looking at where we are pretty much today. And, you know, these things are out there. I think that what's really been holding them back mostly is that, you know, consumer resistance to them, you know, the unwillingness to buy them, just the bad publicity. But basically for the future, what we're seeing right now is just the tip of the iceberg. There's a new technology out there and it's called Crispr, and it's a mouthful. Clustered regularly interspaced short palindropic repeats. Okay, so why is CRISPR going to be so much different? It's inexpensive, it's fast, it's relatively easy. I've been told that a high school student, a relatively smart high school student can do this. It's accurate. In other words, it goes in and it does what it's supposed to do, and it's also very precise. It goes to the exact location. So is CRISPR just another tool in your portfolio, like the BT events, the CRY and the VIP? Well, in the same sense that, you know, maybe this is a tool in your toolbox. You know, you've got to think of CRISPR as something that could, you know, travel through that forest on its own with GPS accuracy and pick out that exact tree, cut it off, and take out the bad part of the tree, or maybe even and graft a new part in it. That's you know, an analogy, but that's how accurate CRISPR is. So CRISPR functions at the double helix level, and it can go in here and edit a single base pair. It can, you know, in an entire cell, or, you know, even in an organism, it can go in there and attack, well, we won't call it attack, but it can unzip the helix and change a base pair. And it does that by using a protein, an enzyme here, and you can see where it's unzipping, and, you know, it's going to replace this base pair. So the thing about it is that it's so precise and it's so accurate that it's not going to be any different than any other natural mutation, point mutation, which happen every day in organisms. They happen in our bodies, they happen in plants. And frankly, it's not going to require labeling as a GMO, at least not yet. This is the talk. But then you have the ethics behind it. And this is one of the things that came up with genetic engineering. Now, the difference between CRISPR and genetic engineering also is that you're not bringing a gene splice from a different species in you know, your target species. It's not a transgenic type of exchange. Okay, so, you know, then you've got to start asking yourself, well, why do you stop? You know, because CRISPR can work on virtually any organism on the earth. Why do you stop at just changing the corn. Maybe you could make the corn resistant to the worm, but why don't you make the worm so, it doesn't like corn, or the Crispr event is even fatal and can wipe out the species? Well, then you get into unintended consequences. I mean, then next thing you can do is wipe out raccoons. Maybe change birds. You guys all voted that birds were your number one issue. Change the bird so it doesn't like corn. Take them all out, or just make the guy that shoots them even a better shot. This is where we're headed with CRISPR. It's a technology that's coming at us that is poised to change the whole world, and I can't emphasize that enough. It's maybe one of the biggest events that's happened in mankind's history. And are we going to have a world without pesticides? I don't think so, but it might reduce them a lot. And, the seed is going to be the delivery mechanism for these processes. So, I guess I wanted to go back and look at this time frame again. 10 years looks like a long time, but again, going back to 9/11, 20 years ago it happened. 10 years ago, this was had already been rebuilt. So 10 years is nothing in even our lifetimes. This stuff is coming at us like a freight train, or maybe it's coming at us like this freight train, So I guess we live in a time of miracles here. And like Einstein said, you can look at everything as a miracle or nothing, and I choose to look at it as a miracle that's possibly going to change mankind. But yet, there's the potential for things to go badly. And as Douglas Adams said, never underestimate the ingenuity of complete fools. Chuck, can I turn this back over to you?
You certainly can if you want to stop your share.
Yep.
Okay.
I think I did, right?
Yep. So any specific questions for Blake while it's still fresh?
This is tough giving you guys a talk on Zoom. I've never done that before, but you have no feedback from the audience. But I guess, the one thing that I'd like to emphasize to you a little bit more too is that don't expect the seed companies to keep coming out with an awful lot of BT corns until the public sentiment toward them changes. There's just too much money involved in it. You see companies like Seminis, they have one or two, and that they're looking at. Syngenta has a couple they're looking at. It's not going to be the major player out here in the next few years.
Okay, thanks, Blake. Any questions for Tom? I know Tom's got to leave. He's got another meeting at 2:30, but Tom, I did have a quick question for you, and that is, are you using any of your trap catches to actually time your sprays, or you're just sticking to a preset schedule?
Yeah, that's a great question. Something else that I had mentioned that a group of entomologists, including Summit Cornell, we're putting in a big federal grant to really focused on sweet corn pest management. And some of the research objectives we're going to do is really take a fresh look at trapping and what trap catch means. Look at some of these thresholds again, because the thresholds were developed and things have changed. Insecticide efficacy has changed. Probably our corn earworm populations, they may be overwintering in areas like I had mentioned. So I know I'm not answering the question. I'm saying we're going to be looking at it. I think what my experience in Virginia has been, the whole timing of trying to get trap catch data to that grower in a timely manner has been a challenge. And more so than not, we have such corn earworm pressure that it's probably just best to protect against that insect. To be honest, I think that's what's been going on. Can we improve upon that? We've looked at from a research standpoint. Yeah, you can reduce your number of sprays if you try to follow it, but every time we do, there's a little bit, may not be statistically significant, but if you look at percent clean ears, it drops when you drop off sprays. So we need to take a fresher look at how to use that. I think there's a lot of questions, and what's the best lure, the best trap, how many traps, black lights versus, can we still get black lights? Can we still get black lights where agents are using them or growers use them? That's a great question. I think we need to just take a fresh look at the program and what we can do. And so, yep, that's all I've got for that.
Well, that's all right. And we also know that it could be, the certain cloud rolls over one farm and drops a bunch of them, and the farm 3 miles down the road has none, so I know it's a hit or miss thing, but no, that was perfect. That's what I was hoping for. So if anybody else has any questions for Tom or for Blake, go ahead. At this point, I'll let you unmute yourselves and go ahead and ask away. Well, I hear somebody maybe unclicking. All right. Well, I think at this point, Tom, I greatly appreciate you joining us today. Blake, I also appreciate you joining us today. Great presentations, great information. And I greatly appreciate everything that you did. At this point, I think, Tom, I know you got to get going. If you want to go, that's great. But I'm going to ask everyone else to hold on. And I've got a couple more poll questions here for you to answer, just so that we get you recorded, so that you can go ahead and get your pesticide credits. So hopefully you're seeing the next poll up on the screen. It's when spraying your sweet corn for worms like European corn borer, Cornea worm, or fall armyworm. I usually just spray once a week, no matter what. Talk to fellow growers at market, see what they're doing, use trap catches and scouting reports from my local extension specialist, or I just don't spray. All right, so looks good. We've got, give it another second here or so. So looks like we're about half of you actually look at the scouting reports, which is great. And then a third of you also just spray once a week. And you kind of heard Tom say that that's kind of what their position has been, but hopefully they'll continue to look at that and maybe have some more information for us in the future on how to use those trap catches. All right, so Chelsea, you can end that one. And if you want to put the last one up. So the final poll is, after viewing this webinar, something that I will change about the way I spray my sweet corn is nothing. The way I do it is working. Rotate with different chemistries, quit growing sweet corn, or pay more attention to scouting reports. Great. Looks like the answers are coming in. Almost half say they'll rotate with different chemistries. And then a third are not going to do anything different. Another third are going to pay more attention to scouting reports. And 3% are just going to quit growing sweet corn. So, all right, Chelsea, you can go ahead and close that poll. Again, I want to thank everyone for joining us this afternoon. I greatly appreciate your support. Make sure you go back to that Teachable site. Again, we posted a few resources on that site for you to go ahead and take a look at whenever you have a chance. And with that, I wish everybody the best in 2021 in the growing season. I hope you all have a great rest of the winter and keep my fingers crossed for a good early warm spring.